Good morning or afternoon. My name is Janie Montblanc, and on behalf of the Great Basin Fire Science Exchange, the Native Plant Project, and our partners, I would like to welcome you to this webinar titled Verification of Sagebrush Subspecies from Seed Samples and Finding the Right Place for Successful Restoration, presented by Bryce Richardson with the U.S. Forest Service Rocky Mountain Research Station. And just to let you know, you are on the right webinar. Bryce revised his title a little bit, but the content is the same, I believe. All right, before I introduce our speaker, I will go over some webinar details. If you have questions for the speaker or me, please type them into the questions pane of your control panel located at the top right of your screen at any time during the webinar. I will field the questions for the presenter after the presentation, but it helps us to receive questions throughout the webinar so that we have some ready in the queue. I also want to let you know that whatever you do in your control panel does not affect the webinar presentation. So you're welcome to type a test message or test your audio at any time during the webinar. If you're having problems with your audio, please open your audio pane and check your audio selections. Now I would like to introduce our presenter. Bryce Richardson is a research geneticist with the U.S. Forest Service Rocky Mountain Research Station Shrub Sciences Laboratory in Provo, Utah. His research focus is on shrub and tree ecological genetics with an emphasis on climatic adaptation and population genetics. Currently, his research involves understanding environmental adaptation of big sagebrush to help guide restoration of sagebrush ecosystems. Welcome, Bryce, and thank you for being here today.
Thank you, and good afternoon from Utah.
All right, I am making you the presenter now. All right, I see the like 2-slide version.
Oh,
okay. Or the notes version. Yeah, there we go.
How's that?
Perfect.
All right. So to start off with, I thought I'd talk a little bit about the National Seed Strategy and how it applies to some of the research I'll be discussing. The seed strategy has 4 goals, but 2 I've highlighted here are most relevant to the discussion today about sagebrush. Goal 1 is to ensure the reliable availability of genetically appropriate seed, and goal 3 is to develop tools that enable managers to make timely, informed decisions. So for goal 1, I hope to show that we can make progress in acquiring genetically appropriate seed through seed testing for big sagebrush. And for goal 2, I hope to convince you that we need to make strategic decisions about where to implement sagebrush restoration given climate change. So big sagebrush is composed of 3 predominant subspecies. One subspecies is mountain big sagebrush, Artemisia vaccina. Another subspecies is Artemisia tridentata, or basin big sagebrush. And the third predominant subspecies is Wyoming big sagebrush, Artemisia tridentata wyomingensis. And all these subspecies support a diverse array of wildlife, insects, forbs, and grass species. And often these subspecies support different assemblages of flora and fauna. So why worry? Well, sagebrush has been in decline, and a number of sagebrush obligate species are in peril due to the loss and degradation of sagebrush ecosystems. Principally, degradation is caused by fire, mainly influenced by human disturbance, human causes, invasive plants. Cheatgrass is the main player, but there's others. And all this is influenced by climate change. So early on, back in probably the in the 1950s, people were wondering whether these different forms of sagebrush were actually meaningful. In other words, were they genetic or was it just phenotypic plasticity? So some of the early work showed that growth rates vary among subspecies. Basin big sagebrush has a really high growth rate compared to the other two subspecies. And it was also shown that there's also early root growth differences. And in this case, Wyoming big sagebrush was shown to have early root growth, faster early root growth. There's also chemical differences that is really interesting. Early on, it was discovered that there's different levels of coumarins in sagebrush, and it was discovered a UV fluorescence test is a good way of differentiating basin subspecies from mountain big sagebrush. And there's been some work done on some of the volatile compounds in sagebrush, and there are differences among these subspecies. And of course, some of the studies I've worked with have shown that this also amounts to differences at the DNA level. So how are these subspecies arrayed across the landscape? So in basin ecosystems, you generally see with increased soil depth and moisture an increase in basin big sagebrush. With decreased soil depth and moisture, the environment tends to favor Wyoming big sagebrush. And in montane ecosystems, generally support mountain big sagebrush. But there are ecotones among these different mosaics, and it's been shown that hybridization does occur in these ecotones, in this case between basin big sagebrush and mountain big sagebrush. And they've shown that these hybrid areas, if they take the hybrids outside of this ecotone, they perform more poorly compared to the parental subspecies. So this is a good photo to kind of illustrate these differences. Here you can see that we have a draw, and in this draw we have the conditions, enough soil depth and moisture to support basin big sagebrush. But as soon as you leave this microclimate and get up into slopes and the uplands, soil depth changes and it can only support Wyoming big sagebrush. So the messages for restoration and wildland seed collection are placing the correct subspecies in the correct habitat is critical to the success of sagebrush restoration. And if you're collecting wildland seed, the more topography, heterogeneity in the landscape, the more likely you are to find a diverse community of different subspecies of sagebrush and potentially hybrids as well. So for this seminar series, I thought I'd put this in kind of the context of our mantra, which is the right seed in the right place at the right time. So for sagebrush, the right seed needs to focus on both making sure we have the correct subspecies and also putting it in an area that's adapted to, so the correct seed zone. And for the right place, we need to look at placing the subspecies in the correct habitat but also long term we need to start taking into account climate change and how will climates, and the question would be whether climate will support sagebrush for the upcoming decades. And the right time, of course, kind of revolves around weather, and there's already been some good talks on how weather affects establishment of sagebrush and other plants. So in bold are kind of the two topics I'm going to address today, both looking at tests to determine subspecies and also how climate change might affect the distribution of big sagebrush in the future. So first, I'd like to talk about some subspecies verification studies that we've been working on using seed. So the research goals were to determine if seed weight differences exist between subspecies. And probably the most important part of this is determining if there's differences between Wyoming and Basin big sagebrush. In the study, I'd like to assess how the environment, genetic by environment affects seed weight, and then use the confidence intervals around these means to evaluate commercially collected seed. And finally, what we're currently working on is developing a protocol for seed testing, and then compare the results between different seed labs. So this study got started, well, it came out of seed collections for common gardens, and then the seed collected from these common gardens were also used in this study. So the two common gardens that we did collect seed from that are used in the study is Majors Flat, Utah, and Orchard, Idaho. We were unable to use another site close to Majors Flat because the majority of the plants had died after the first and second year. So I'd also like to highlight the different environmental conditions between these two common gardens. Majors Flat is a mountain big sagebrush site, higher elevation, receives on average about 20 inches of rain per year, compared to Orchard, which is a dry basin site, which received about half the precipitation that Majors Flat received. So this is kind of the breakdown of the seed sources and locations that went into the study. I have things grouped out into both subspecies and cytotype. So that's the ploidy level, and it can vary within subspecies except for Wyoming big sagebrush that is from all the samples that have been tested over the years, it's always been tetraploid. And I'd also like to point out that not all the seed sources were represented in each garden because a few of these sources did not produce seeds in the gardens. So this is just kind of a timeline of the seed collection and sampling for this study. So in 2009, seeds were collected from the wild from the seed sources, and these are what I refer to as the original seed. And in spring of 2010, these collections were planted at the common gardens. And then in 2012 and 2013, seed was harvested from these common gardens. And you can see the bags on these inflorescences, and that's what we use to collect the seeds. And finally, this data was used to compare with the BLM seed lots, which were collected in 2013 and 2014. So from the gardens, we sampled 2 plants per population, which had 2 bags on each plant, and then a total of 6 weighs per plant. And this was done in the 2 gardens and for 2 years. So we had a total of over 1,400 sample weighs. And a sample weigh is basically 10 randomly selected seeds that are weighed. And in the original collection, we used 5 plants per population, and these were weighed 3 times per plant, giving a total of 567 sample weighs. So for the analysis, we used a linear mixed effects model, and the advantage of this type of analysis is you're able to partition the genetic variation from the environmental and G by E variation. So the fixed effects were assigned to the genetic parameters, which in this case are subspecies and cytotype groups. And then the random effects were assigned to the environmental and G by E features, which were basically the experimental design. This included the gardens and the original collection and the year, and along with G by E effects, environment by population, and environment by population with family nested within it. So a little bit about the results. This is kind of an overview of what the seed weights look like from using box plots. And these box plots are organized by collection or garden. So we have majors, orchard, and the original. So just a few observations from this. The groups were relatively stable across environments, especially for basin big sagebrush. We see very little environmental variability among the subspecies. We saw the most with tetraploid bassian or mountain big sagebrush. And overall, we see a slight increase in the groups at majors flat compared to orchard and the original collection. So this is just kind of the breakdown of the mixed model results. I guess 2 things I want to point out in this slide is we do have highly significant environmental effects and highly significant fixed effects or genetic effects. But if you compare the level of variance in the random effects terms compared to the estimates from the fixed effects, you see quite a big difference. So these are rather low, and the differences between these different subspecies cytotype groups is rather large. So in this case, for basin big sagebrush, the estimate would be it weighs about 1.76 milligrams. And then you'd add, for instance, almost 1 milligram more to get to the estimate of Wyoming big sagebrush. So just to dig a little deeper into the environmental effects, this is kind of a dot plot showing the year by garden effects for different years and different collections. So you have year 2012 at Majors Flat and year 2013 at Majors Flat for different subspecies and cytotype groups. So the dot is the random effects term, and then the whiskers are the standard error for each subspecies cytotype group. So there's some patterns I'd like to point out. One is we see a really high positive effect at Majors Flat in 2012, but in 2013 it goes way down. And then most of the standards errors overlap among the different gardens and years. But just to put this into perspective, even a plus or minus difference of 0.2 milligrams is rather small, considering Wyoming big sagebrush still weighs about a milligram more than basin big sagebrush. So why was there such a big drop, especially at Majors Flat between 2012 and 2013? I looked at the weather data, had similar temperatures and precipitation, but one thing that happened was at this site especially is because of the increased precipitation, there was rapid growth, and the rows were actually closed by 2013. So I think what we're seeing is really an effect of competition. And I'm not going to show this, but if you look at seed yield too, matches this, or is even more dramatic. Seed yield really drops between 2012 and 2013 at Majors Flat. So this is the confidence intervals built from the model. And I guess two things to point out is Basin big sagebrush and Wyoming big sagebrush, the confidence intervals, even at 99%, do not overlap. And we expect seed lots with seed weights greater than 2.3 milligrams to have a high proportion of Wyoming big sagebrush. So if we take those means or estimates and the confidence intervals and compare them to the commercial seed lots, we see the kind of combined overall look at those in this plot to your right. So what we have is the darker gray distributions are the weights of Wyoming seed lots purchased by the BLM in 2012 and 13 compared to the confidence intervals based on the experimental data. So the dashed lines are the 99% confidence intervals, and the solid lines is the mean. The red would be, of course, Basin big sagebrush, and the blue would be Wyoming big sagebrush. So based on the means, we saw that 17% of the seed lots from that were labeled as Wyomingensis met the minimum weight or the lower confidence interval of the experimental data. Although I'd like to point out that a lot of these others, all the other seed lots appeared to be a mixture of both Wyoming and Basin. So the other thing we did was we took 4 of the seed lots and germinated some of the seeds and did flow cytometry. and we took some that met met the lower limits of the confidence intervals, which is seed lots A and C, and another two that did not, which was seed lots B and D. And we found, once we did flow cytometry, that for A, the percentage of tetraploid plants was high, it was 97%. C, which was similar, maybe a little lower in its distribution, had 57% tetraploid plants, whereas B and D had 11 and 0. Let's see. Okay. So, in summary, environment does have an effect on seed weight across these extreme climatic differences between gardens. And it also appears that rapidly growing plants with ample water produce slightly heavier seeds. However, these effects do not blur the seed weight differences between Basin and Wyoming big sagebrush, indicated by the confidence intervals. And in terms of genetics, seed weight is a strongly controlled genetic trait, and it appears the most important factor in determining the seed weight is cytotype or polyploidy. So a few recommendations for seed vendors. Obviously, you're never going to be able to get a seed lot that's 100% Wyoming big sagebrush. There's just too many small mosaics on the landscape, in my opinion. However, I think site choice is going to be key in obtaining higher proportion of Wyomingensis or Wyoming big sagebrush seed. And I think it only takes a few plants of Basin big sagebrush to throw off the weights. And that's because the amount of seed produced by Basin big sagebrush compared to Wyoming big sagebrush is quite different, especially we consider when they grow in different habitats. You're looking at, in our gardens, we saw about a 10-fold difference in seed yield between these two subspecies, and I expect in the wild the yields would be even higher. So for seed purchasing agencies, seed weight can be used as a diagnostic test to evaluate the proportion of Basin and Wyoming big sagebrush in seed lots. This, along with a UV test, can discern mountain big sagebrush from these Basin subspecies. So this year we've been developing a seed testing protocol, and the goal is to develop a standardized procedure for testing subspecies that fits within the established seed testing guidelines. And the test we're proposing is a UV fluorescence to determine if seed lots are composed of mountain big sagebrush, seed weights from seed lots based on a random sample of 8 replicates of 100 seeds, and for this year, flow cytometry to determine ploidy on about 40 to 80 seedlings from each seed lot. So this will be done using the Oregon and Idaho seed labs, along with some testing at our lab here. And the goal, of course, will be to compare the results from each lab for the UV test and the seed weights, and then to make recommendations to the BLM and seed vendors. Performing flow cytometry from germinated seeds, we're hoping will help compare or associate seed weight with the proportion of diploid and tetraploid plants in the seed lot. So, the question has come up, why don't we just do flow cytometry since it's so definitive? And I guess the problem with that is if there's a fire year and there's very little time between collecting the seed and getting it on the ground, and flow cytometry is probably going to take at least a month to get the data back. So if it's not a fire year, I think it would be okay, but because of that, I think it's going to be important to associate the proportion of 2x diploid and tetraploid plants with the weights that we observe from the same seed lots. And I think this will also help guide where we put the plants on the landscape. For instance, for seed lot A that has a high proportion of Wyomingensis, it would ideally go on a very dry, homogeneous site that's, predominantly supports Wyoming big sagebrush. Whereas seed lot B would be more suitable for sites with higher topographic heterogeneity, where you see conditions like in the photo, a combination of both basin, Wyoming, sagebrush. So now I'd like to just kind of shift gears a little bit and talk about climate and climate change and its impact on sagebrush. So the question is, how do we identify areas that pose the most risk for sagebrush restoration? And obviously, weeds and fire frequency are major factors that we need to consider. But I think we also need to start thinking down the road about climate change and what is the right place for sagebrush to be resilient for the next few decades. And one way of addressing this question is through bioclimatic models. And a bioclimatic model is just using presence and absence data of a particular species or even an ecoregion to assess the climatic patterns or parameters, that govern the distribution of a species. And then these climate variables that are important in the prediction, which are referred to as the model, are projected, are used to project the predicted species distribution, or what we refer to as the climatic niche. And these models can be projected for both contemporary climates and also future greenhouse emission scenarios. So some recent work that was done by Rayfeld et al. That looked at the desert biomes of the western North America. And in the map on the left shows kind of the four deserts, both the Great Basin, the Mojave, the Sonoran, and the Chihuahuan. But what I thought I'd show is how the Mojave Desert changes over the next several decades. And it shows that by 2060, the Mojave starts to expand northward up further into western Nevada, and also the climate is suitable for Mojave-like conditions even up into the Columbia Basin. So we can also, of course, do bioclimatic modeling on species or even subspecies. In this case, we did a study on modeling Wyoming big sagebrush. So our approach was to use 131 presence points of Wyoming big sagebrush, and the majority of these were all confirmed through flow cytometry or chromosome counts. And then for absence points, we use species with allopatric ranges to Wyoming big sagebrush. So we used blackbrush, which is an ecotonal species between the Mojave and the cold deserts, mountain mahogany, which is a higher elevation tree, and a few others. And then we projected this climatic niche model for Wyoming big sagebrush for contemporary climate and also decade 2060 using kind of a moderate greenhouse gas emissions scenario, which is representative RCP 6. So the map is kind of a composite of both the current and the 2060 projections. So in the yellow is basically the current projection of Wyoming big sagebrush, but in this case it also is showing where areas contract. The gray shows stable areas for Wyoming big sagebrush, and the blue predicted expansion. And overall, we see about a 38% reduction in Wyoming big sagebrush climatic niche. And the other thing we did was we looked at, in the Great Basin, what kind of climate variables were contributing to this predicted reduction. And of all these variables, annual dryness index, which is an interaction between warm temperatures and precipitation, had the most significant differences between stable and contracting areas in the Great Basin. So in this case, contracting had a higher annual dryness index than stable. But I think this also kind of supports the idea that if it's getting warmer and drier, that it also decreases the frequency of favorable climatic or weather events that would support the establishment of Wyoming big sagebrush. So the other thing I did was we've also done bioclimatic modeling on blackbrush, and it's interesting to compare these two taxa. We can see their contemporary distribution on the left, and in 2060 we can see how when blackbrush, this ecotonal species, moved northward into Nevada, the Snake River Plain, and even the Columbia Basin, which supports the paper by Rayfield showing that the Mojave Desert is expected to expand northward. One caveat to this is, of course, these models are climate-based and they don't take into account soils. And of course, a lot of the area in western Nevada is salt desert, and obviously blackbrush doesn't tolerate salt. So some of these areas, are not a fit for blackbrush, and I think we're going to have to consider other species. So with that, I think we need to start looking outside of the Great Basin ecoregion to some of these ecotonal areas for potential sources of shrubs to plant in the lowest areas of the Great Basin, lowest elevations. So some of these shrubs that might be good candidates for seeding in some of these areas that are expected to lose sagebrush, or potentially black sagebrush, blackbrush, winterfat, some of the rabbitbrush species, of course Atriplex species, and ephedra. So in summary, changing climates will alter the distribution of our desert shrubs. A significant portion of the cold desert that currently supports Wyoming big sagebrush will likely favor warmer ecotonal species like blackbrush in the future. And in the Great Basin, this shift will likely result in a warmer and drier conditions that no longer favor sagebrush establishment. I think research needs to continue to assess shrubs, grasses, and forbs that are adapted to warmer and drier conditions to meet these changes. And I think, of course, the shrub communities in these areas will only persist if we move the correct shrubs or plants into those areas. This change is too fast for them to keep pace. So just a few final thoughts. I think to provide a fighting chance for sagebrush, just better establishment and resiliency for this species, we need to identify the species composition of seed lots and then use this information to tailor this composition to the habitat or the site where restoration is needed. And I think this needs to be done with natives that have co-evolved with sagebrush. And I think we need to start prioritizing restoration efforts to areas that will support sagebrush for decades. With that, I'd just like to acknowledge my collaborators and assistance from a number of organizations and people, and my funding sources. Thank you.
Great, thank you so much, Bryce. All right, if you have questions for Bryce, please type them into the questions pane of your control panel at the top right of your screen. The first question is from Molly Boyder, and she asks, for those regions proposed to lose Wyoming sagebrush, Columbia Basin specifically, what is the recourse? Should we be focusing on using Artemisia tridentata wyomingensis for restoration, or abandon that for blackbrush or other species?
I think for your particular area, I think we still have to make an effort for the time being, to try to restore sagebrush. But hopefully, my plan, hopefully other researchers will start looking into some of these other shrubs like blackbrush and test them in more northerly climates to understand how they're going to, test their fitness, basically. But yeah, I think, over the long run, we need to start kind of mentally preparing ourselves to look elsewhere.
Are you aware of studies right now that are trying to plant these shrubs and see how they do in these areas?
We actually are planning on planting, or kind of common garden trial of blackbrush, where we're going to put it up in the Snake River Plain, along with some sites in the Mojave, and see how it does. But I think, you know, that can be done with a lot of other plants as well.
Interesting. Thank you. Okay, well, so far there are no other questions. Oh, here we go. Horsebrush grows in alkaline soils in the Columbia Basin. I don't know if that was a question or just a statement. Statement.
Obviously, if there's, you know, some plants are going to be have a greater tolerance for climate change than others, and if those plants can be found in those particular regions, then I think we got to use those. But I'm sure there's going to be situations where we're going to have to move our target south to find the right plants. South, or, you know, in warmer conditions.
Great, thanks. Jessica Moran asks, how does cattle in the Great Basin affect Wyoming sagebrush communities?
I'm sure it's not good. You'd have to ask somebody that's expert on grazing. But my opinion is, you know, it's probably a detriment.
Okay, thanks. All right. Well, you can, if you still have questions, you can type them in. But while we're waiting, I can just let you know that at the end of this webinar, there will be a 3-question survey. So if you wouldn't mind answering that we'd really appreciate it. And I will post the recording of this webinar on our Great Basin Fire Science YouTube channel this afternoon, and the link will be automatically sent to you through the GoToWebinar system tomorrow. Our next webinar, titled The Incredible Diversity of Sagebrush Chemistry and Its Potential Value in Restoration, will take place next month on March 15th. If you have further questions regarding this webinar or have requests for future webinars, please email or call me at any time. Okay, well, I guess that was indeed the last question. So thank you all for your participation today, and thank you so much, Bryce, for your presentation.
You're welcome. Thank you.
All right, have a great day, everybody.